Hey, boys and girls, we're ready for section 4. Maybe. What do you think? Are we?
I was born ready for it.
What's happening? Or another.
Yeah. Okay. So, section 4 of the paper, Hot Spring Origin of Life. So I guess we want to start reading and try to figure out what this section. Now this section goes through, I think, the 7 steps that are they outlined in here? No, it was somewhere else. Hang on a second, I'll find it. I'll get us an illustration, but you may want to go ahead and start reading. What do you think?
I'll go ahead.
Yep.
Okay, so integrating the chemical, combinatorial, and adaptive scenario in a geological setting. I bet Orge wishes he was here right now. Any scenario describing the origin of life must first escape the bounds of a working hypothesis into laboratory testing, followed by further testing in natural conditions. For instance, laboratory simulations have inspired experiments in volcanic hydrothermal fields in Kamchatka, Russia, Bumpus Hell at Lassen National Park, California, Yellowstone National Park in the United States, and most recently in Rotorua, New Zealand. The results of these and the Rotorua studies are reported by Deemer et al. 2019. With some confidence, we can now integrate the chemical,
We're working on it, dude.
Combinatorial and adaptive aspects of our scenario within a macro scale environment meant to represent a Hadean landscape. I love it that they're calling the original, primitive landscape hell.
Basically.
Yeah.
Figure 7 depicts this landscape, and the 7 specific stages we propose can lead to the origin of the first microbial communities. Each stage has a number, and we proceed through them one by one to describe how our scenario, in which cycling hot spring pools play a central role, integrates physical, chemical, and combinatorial processes within a prebiotic geological landscape.
Okay.
5.1.
So,
sure.
So I wanted to do this first. Share screen. Application window. So this is figure 7.
Cool.
So, yeah.
There we go.
So we've got synthesis, number 1. That's the first section. And they go through each of these sections: that accumulation, concentration, distribution, adaptation, and colonization. And I think they make a case for, like, if this had happened in basically these smokers in the ocean, that accumulation would have been a problem. That the concentration of the biochemicals would have been a problem, and that's kind of where they came onto this this theory of the hot spring hypothesis. But we will. It'd be cool if you could pull up that illustration, immutable, and display it, and then we could switch back and forth if we have to, because I can only have one screen at a time, because StreamYard, as we know, is kind of retarded.
Yeah, I agree with that one.
Yep, they don't really care about their customers very much.
And you're like the most loyal customer.
I know. I give them money. Okay.
That's why we need to go into a new. That's why we need to go to a new platform, like,
OBS and Zoom.
Streamlabs.
OBS would solve the problem, but anyway, we'll get there. 5.1.1 synthesis.
The setting is the Hadean. I'm sorry, are you reading? Sure,
no,
go
no.
ahead.
No, I'm not. No.
Oh, okay. The setting is the Hadean Earth approximately 4 billion years ago, by which time a global ocean has formed with emerging volcanic landmasses. The solar system is a dusty place with its accretion disk still being swept by young planets and large asteroids that experience numerous collisions. These dust particles are coated with a thin layer of ice containing organic compounds synthesized earlier in their history, along with the organic compounds adhering to their interior porous mineral matrix. This organic material was delivered to Earth's surface as an abundant accretionary infall that continues at a reduced level today. So we're talking about really quite a high volume. Think of all the water that had to come onto Earth from comets and dust and everything within what, 500,000 years?
500 million.
I'm sorry, 500 million. Yeah. Right. I meant to say that. I don't know why I said 1,000. This organic material was delivered to Earth's surface as abundant accretionary infall that continues at a reduced level today. Atmospheric photochemistry and volcanic geochemistry also contributed organic compounds to the available organic inventory. Wu and Sutherland, 2019. Separation of organic meteoric compounds such as amphiphiles and monomers will begin as soon as dust particles and meteorites come into contact with acidic atmospheric moisture, delivering larger organic complexes designated A in the figure 7 on the left side of the bar of chemical complexity.
Yep. And for some reason, I don't have Wu. Okay.
You don't have Wu?
Wu. I don't have Wu.
Well, it's good not to have
Wu. So indeed, we don't have Wu yet.
I had Wu.
This is a Wu-less podcast or broadcast.
Well, wait a minute. I had Wu. I didn't download the Wu. I downloaded the Wu paper, but Parker paper is interesting, and that's the one that talks about. I think. Hang on a second. Parker, 1982. Nope, that's something else. Sorry. Shut up. I'll shut up now. Okay. So anyway, there was a massive inflow of material from outer space during this 500 million years, and I was thinking, like billions of tons or something like that, but it was significantly bigger than that. It was kind of amazing when I looked at some of the calculations about the amount of material that came in. And out of that, we know that there were lots of biomolecules and all of that stuff. So lots of starters. So anyway, so on to the accumulation phase, which is number 2.
5.1.2. If this material falls into the ocean or larger bodies of water on land, it will be dispersed into a dilute solution that cannot support chemical reactions. However, if these organic compounds accumulate on volcanic land masses, they become available to participate in cycles of significant chemical reactions. The initial collection points are mineral surfaces similar to those present in volcanic sites today. For instance, rough or vascular surfaces of basaltic lavas can collect solutions. Small pools also form when clay particles or precipitated silicate materials seal otherwise porous surfaces. The mineral edges of pools, which may have moist sediments, can preferentially capture windblown dust laden with these compounds. Hydration driven by precipitation, dew, humidity changes, or deliquescence can then release organics and begin to promote the condensation reactions described in section 1.
Okay, and then we go on to the next page, I believe. And let me, kind of hard to hit that little thing.
Speed, you want to go ahead and read that, or immutable?
Okay, so immutable.
Yep, okay. 5.1.3, concentration. Assuming that amphiphilic compounds are present in the mixture, the reactants will be encapsulated and concentrated within vesicular compartments. Because hydrothermal field pools are interconnected, chemically complex, and vary widely in temperature and pH, products formed in one pool can mix with different compounds formed in neighboring pools. And so, chemical processing is not limited to a single pool having one set of conditions. Examples of such pools are found today at hydrothermal fields in Iceland, on Mount Minotsky in Kamchatka, Russia.
Mutnovsky.
Mutnovsky in Kamchatka, Russia. Mount Lassen on Yellowstone National Parks in the United States, and Rotorua in New Zealand. The association of these products within membranous structures keeps them in a concentrated state regardless of the hydration and dilution of the pool, and sets the stage for combinatorial selection to occur in a cycling environment.
Okay.
5.1.4 Cycling.
So concentration. Go ahead.
Hydrothermal pools undergo wet-dry cycles resulting from precipitation and fluctuating water levels driven by active hot springs and geysers. The large circle, introduced first in Figure 5, is set in one such pool that has a bathtub ring, quote-unquote, of dry solutes around the mineral edge. Polymers are synthesized by condensation reactions occurring within these dried films, which form a multi-lamellar matrix. During the hydration phase. 2, vesicles bud off, encapsulating systems of polymers to form protocells. 3, a partially hydrated gel phase of crowded protocells. 4, forms an interactive aggregate within an increasingly concentrated solution and then undergoes drying and fusion into a multilamellar matrix. This process generates random sets of polymers captured in vesicles to form vast numbers of protocells. Frequent cycling of these populations initiates the combinatorial selection process that drives chemical evolution and enables the emergence of ever more robust protocells. As the protocells continue to undergo the stresses of cycling, most will be disrupted, their compartments leaking out or dispersing through disrupted membranes, but a rare few actually, are likely to contain polymers that enhance their survival. Some protocells are successful natural experiments that survive testing during the hydrated phase, but they cannot express catalytic functions nor live independent lives. For the system to evolve, it must engage in a network in which properties and innovations of molecular selection are shared. Here, the gel phase of the cycle can play a role because the close contacts and fusion events within the aggregate allow the sharing of polymers that have beneficial functions, particularly to the ability to create useful metabolic products and catalyze polymerization and replication. At this point, the system has reached a form of pre-life, B in Figure 7, on the left sidebar. If this seems implausible, it is worth recalling research results from Phil Holliger's group, in which it was reported that ribozymes evolved in the laboratory by selection and amplification were able to catalyze the polymerization of RNA molecules nearly 200 nucleotides in length. Similar results were reported more recently by Horning and Joyce, 2016. The products of selection that cycling systems generate can lead to the stepwise emergence of increasingly functional polymers described in Section 3. The aggregated protocells in the gel phase resembled the progenote concept by Woese and Fox, 1977. And
we're getting there.
And Woese, 1988. Progenotes represent a transitional form between simple chemical systems and the more complex biochemical functions of biology. It is interesting that progenote evolution bears a certain analogy to booting up the computer's operating system, in which services start up, share, and support higher-level applications.
Uh-oh.
Again with the analogies. The programs of life are polymer systems selected to perform primitive versions of biological functions, capturing nutrients and chemical energy from the environment, metabolically alter the nutrients into useful compounds, growing by polymerization of monomers, and catalyze replication of certain of those polymers. Eventually, these systems no longer depend on the scaffold of hydration-dehydration cycles for the chemical potential driving polymer synthesis. Instead, individual protocells Individual protocells will begin to utilize polymer catalysis to perform these tasks and must therefore pass on complete molecular tool sets that include primitive genetic instructions to guide their synthesis. The evolutionary chasm to life will be crossed when protocells begin to divide into daughter cells, each receiving viable sets of these catalysts and genetic instructions.
Okay.
Speciation and specialization of living cells then cross into the Darwinian threshold, described by Woese 2002. The tangled roots of the horizontal sharing through communal progenotes now supports the growth of multiple interwoven branches as the tree of life continues to evolve. By 3.5 billion years ago, a common ancestor has emerged with a genetic code and translation system, leaving behind telltale signatures of stromatolites and microfossils preserved so well in ancient marine, lakeshore, and freshwater hot spring settings in the Pilbara region of Western Australia. Distribution.
Okay. Well, let's not go there yet. So that's about 1 billion years, I think, is what they give themselves, right? Like 4.5 billion to 3.5 billion years ago. And Bruce Damer has I'm going to put links to all the papers that are referenced here. I think I got them all. I'm not sure if I got Walter et al. But anyway, I have most of them. And he makes a wonderful video, a couple wonderful videos about how this hot spring idea got started. And Damer and Damer both are working on a paper that they haven't published yet, where they actually did this in a laboratory and they have results. And he has some videos on that. So all of that is there, and all of this stuff is backed up by each of these papers. So it isn't just like assuming things or imagining things or making a story. It's just that this paper sort of brings it all together. And there's a whole lot of work underneath this that needs to be looked at. And I am going to put links to all of those papers, all the ones I have, in the description for interested parties. So, okay, now we're on to 5.
Okay, 5.1.5, distribution. Any hypothesis for the origin of life must propose a pathway for distribution. If the cyclic processes described above occurred in an isolated pool in which a robust progenote population happened to emerge, there would be no way for it to escape this potentially short-lived pool and continue to evolve and adapt to new niches. In interconnected hot spring pools on mountainous volcanic landscapes, Interconnected hot spring pools on mountainous volcanic landscapes have the obvious advantage that there is a continuous downhill flow from pool to pool driven by gravity, so that protocell populations can spread to other pools with distinct characteristics related to temperature, pH, ionic content, and mineral composition. There, they will be subjected to additional stresses, driving them uphill in complexity, ultimately to the division of protocells into living cells and the emergence of the primitive early microbial communities. Dehydrated films can also undergo windborne distribution, a mechanism still used today by microbial and algal mat communities. These films are an effective preservation and dispersal mechanism for systems of polymers that would otherwise be degraded by long-term exposure to hydrolysis. Seeds and spores are blown by wind and preserve their genetic polymers against water activity between generations of plants and certain microbial populations today. As protocell and then early living cellular populations become physically distributed across the landscape, they are exposed to a range of conditions that drive increasing robustness to multiple stresses. Innovations that developed in one pool can be shared with populations occupying another. This
process has to wait a minute. Okay.
This process eventually produces tolerance to the extreme environment of salty seawater, thereby providing access for early life to the most extensive but often turbulent tidal marine shorelines. Such an adaptive gradient is observed in the hot springs of Yellowstone National Park today. Thermophilic microbial communities that are chemotrophic use the energy and chemicals available in the upwelling high-temperature spring while further downhill in the cooler and more dilute outflow, photosynthetic organisms capture sunlight as an energy source. Beginning with the first generation of stabilizing polymers, the stepwise evolution of more complex polymer systems would continue to depend on a partnership of polymers with the membranous boundaries of protocells. For instance, Groen et al. 2012 reported that polycyclic aromatic hydrocarbons, PAHs, components of meteoric organics, have a stabilizing effect on fatty acid membranes similar to the effect of another polycyclic compound, cholesterol, on membranes today. Black et al. 2013 showed that adenine has a stabilizing effect on fatty acid membranes, and Cornell et al. 2019 observed similar effects of certain amino acids. For an origin of life on a terrestrial world possessing liquid water, it is the exposed land surfaces that provide the richest array of compounds, environments, cycling systems, and the greatest range of energy sources. Diemer and Weber, 2010.
Let's move up. Okay.
Distribution of progenotes into cooler, watery environments such as dilute streams or lakes leads to a major selective hurdle. Regular wet-dry cycling is no longer available as an energy source, and potential nutrients drop to much lower concentrations. Progenote communities fail until selection discovers some form of photoautotrophy, supporting a much more active, energy-coupled metabolism. Much more complex molecular machines would be required at this stage, including a genetic code driving a primitive version of the ribosome, catalyzed duplication of genetic information, and cell division. It therefore seems reasonable to assume that the transition from passive protocells into more active and autonomous living cells would occur as populations become distributed away from the hot spring environment at C in Figure 7.
Okay. So adaptation, I'll go ahead and try this. Autotrophic microbial colonies ultimately encounter the increasingly brackish water of the estuaries and then the high salt and divalent cation concentrations of seawater. The microbial communities respond by developing robust means to exchange intracellular sodium chloride for potassium chloride and actively transport toxic divalent calcium ions out of the cells. This adaptation requires complex energy-dependent transport of ions by membrane-bound enzymes, suggesting that the first organisms able to live in seawater would have advanced far from the initial Goldilocks conditions required at the site of life's origins. And that is, Mulcadejanian. Anyway, et al. 212. Colonization. And increasingly, now this is the interesting, I think this is what he talks about in this video, as increasingly robust microbial communities arrive at even harsher tidal marine margins, another set of stresses challenges them to adapt. To respond to increased osmotic stresses and shear forces, the microorganisms must develop durable cell walls and an extracellular matrix composed of polymers that strengthen adhesion between individual cells and communal aggregates. Now, I have been reading quite a bit about these extracellular matrices, and there is a strong evolutionary pattern or phylogenetic pattern in lamins and laminins and all of these different intermediate filaments that form these extracellular matrices that go all the way back to very, very early archaea and prokaryotes. And so that is an interesting story all by itself. It looks like some of these proteins are, 3 billion years old and have evolved into many families in modern life. So that is interesting. But that would be the strengthening of cell walls and the ability to handle a little bit of buffering, that sort of thing, or buffeting. The aggregates cling. Did I read that? Yeah. Okay. The aggregates cling to mineral surfaces of pools, and shorelines that are washed by tides 10 times higher than those of today's oceans. Now that's interesting.
That is.
The adhesive mucus, and that washes a lot back into the ocean, that sort of thing. The adhesive mucus that allows the cells to remain fixed on mineral surfaces also binds and cements together mineral grains and sediments, settling over them. To continue to have access to sunlight, microorganisms composing those microbial mats migrate upward and grow new colonies on the top of the cemented substructure. Over many years, multiple layers of microbial films and cemented grains produce the laminations characteristic of stromatolites, which dominate the earliest fossil record and are still forming today in freshwater and shallow marine environments, such as the Hamlin Pool at Shark Bay, Western Australia. At this stage, life would have gained access not only to aqueous environments on the land, but also to extensive marine margins of volcanic land masses and later the ocean volume itself, as cyanobacteria colonized the globe. So this is kind of like the whole story behind this paper, this one section, section 4. And let's see. Now, bothers me I'm missing that Walker paper. But I don't know. We could go on to section 5, but we're just reading. And I'm not sure how powerful and interesting that is. But I mean, what do you guys' thoughts on this?
I think we'll leave it for another day when we have a more when we're able to understand it and talk about it at a. So that way we're able to talk about it intelligibly.
Yeah. So here's the illustration again: the synthesis, accumulation, concentration, distribution, and then the most important part is 6. Okay, that's where we probably take the most flack as to how these extracellular matrices formed and how all of these enzymes formed and how they adapted. And there was a mess of quadrillions of different little test tubes merging with each other, eating each other, absorbing each other, and dividing again and forming different combinations. And so there are all these different tests that basically created very, very small ribozymes and protein enzymes that I think small molecule biology is where we're at here. And that's where we're going to see the most progress made in these small molecules, 20 to 200, well, actually maybe less than 100 amino acids and less than 100 nucleotides. I think that's the interesting stuff. Now there is, let's see, I wonder which paper. Let's see, Nestle mentioned Wosa's paper. He actually has 3 papers. The first one is, where is it? Why? Let's see, is this Wos? Where are you, Wos? I can't find Wos. In a second here.
Or would it be Vos, because maybe he's German?
Wos? I don't know what the hell he is. Wosa? Not sure. I have too many monitors. It's here somewhere.
No, you can't have too many monitors.
I had too many monitors. What is happening?
You need another monitor to help you track all the other monitors.
Oh, I know what's going on. This goddamn thing. At some point, it quits opening papers for me because there are too many papers opened. So it just shuts down on me.
How many papers? Is it like Adobe? Can Adobe only open so many papers?
I think it's like 20 papers that will allow me to open. That sort of thing. Let me test that theory. I just closed one. Nope, that theory ain't working. I am not finding this paper.
It's either a false negative or it's not working.
Not working. My theory's not working. Well, in the meantime, let's take a look at Parker. So, Char. Okay. So there's a paper. This one is removal of organic and inorganic matter from Antarctic lakes by aerial escape of blue-green algal maps. So this was referenced in the distribution section, I think. And that's probably not the most interesting one I have. But benthic algal mats dominated by the prokaryotic Formidium frigidum, whatever. That's an interesting little piece. Well, let's just go ahead and see what I got here. Okay. Right here. Some interesting stuff in the middle of this paper was the talk about stabilizing these protocells. And one of the papers is this, Black 2013. Nucleobases bind to and stabilize aggregates of prebiotic amphiphile, providing a viable mechanism for the emergence of protocells.
Co-authored by David Deemer.
Oh, yeah. And a bunch of other people. Yep.
It was a collaborative project.
Yeah, look at that, xanthine, hypoxanthine. I mean, the complaint by Cell et al. And Change Tan, or is that her name? Change Tan? Tang? I don't know. Tang, Change, whatever. And Stadler, their complaint was that there were all these isomers that were created, right? Well, and then when I look, I find all of these isomers are still active in life today. And some of those, the other thing he talks about is the formation of tars and how that's a problem. Well, xanthine and some of these element or molecules are exactly what those tars are made of. So, I mean, there's all of these different complex isomers that are not homochiral, like they are in today's life, that actually have effects of stabilizing lipid membranes and having, you know, some enzymatic function. So, I mean, when they claim that that's a problem for life, that is the solution for abiogenesis, not a problem. The fact that there were all of these different compounds formed.
Right.
Decanoic acid, that's interesting. That's the one that Deemer threw into that hot spring volcano. Or that little pool, that hot spring that he came up with. Anyway, that was, let's see what else we got here. Altered pH of vesicle transition. So there's a lot of stuff here about lipid membrane stabilization. Shows their methodologies. And that's about it for that paper. And that paper references other papers, and we could go down that hole, but we won't. We're not going to do that. So what else, guys? What other papers are interesting? We have Campbell. And Campbell was from Prebiotic Condensate. Oh, this is cool. Are you excited about these papers I'm showing? Doesn't sound like
very much so. Very much so.
I mean, you don't seem excited. Prebiotic condensation through wet-dry cycling regulated by deliquescence. Deliquescence.
I don't know what that word means. What is deliquescence?
Well, it sounds like something you'd order at a seafood restaurant, I think. Or maybe.
Well, it reminds me of that group, Evanescence.
Yeah, yeah.
They go to the deli. I don't know. Never mind. I'm being stupid. You need to mute me. Mute me now.
No, I think you got it. I think you actually got it right. I think that's closer than any of us have. Deliquescence. Language. Nobody knows. Word neck. Deliquescence. Wiccanary. Formation of a solution by the absorption of moisture from the air. Oh, yeah. Mutable should have known that. Where are you? He's muted. Fucking immutable's muted. How does that work?
Well, I guess he is mutable.
He is mutable. That's why I call him mutable. But Wet dry cycling is widely regarded as a means of driving condensation reactions under prebiotic conditions to generate mixtures of prospective biopolymers. And what do they say in the paper? They've shown, I think I think it was Campbell or maybe it was Black. They showed that up to 200 amino acid polymers can be generated by this process. So that's quite interesting to me. Where are you? Is Immutable left us?
He might be
At the mall
or something. Yeah,
probably
could be.
Are you at the mall, Immutable? Prebiotic amino acids bind to and stabilize. Okay, here's another one. Cornell prebiotic amino acids bind to and stabilize prebiotic fatty acid membranes. One of James Tour's criticisms is that you have to have all these complicated phospholipids to create a membrane. And these papers, are 2 or 3 papers in this group, show that you do not, that rather simple bilipid membranes can be stabilized in different ways with amino acids, or on the last paper, what they mentioned, something else.
Yeah, it's almost like Tour. If I'm understanding things right, I really haven't gone deep into Tour's papers or anything like that. But it seems like he says everything has to be pristine in order to put together the molecules for life or what have you. But a lot of these molecules come pre-made from, the accretionary material. So it's like, okay, here we have the pieces for the recipe. We have the dash of this and the cup of that and the teaspoon of this, and then the hot springs and other processes provide the energy. So it's hard to see really what his objection is.
Oh, we've gotten invited to a conference this week. I have to check my email and make sure I get that information. And I also, Jerry, I do have to reach out to Bruce. I'm going to try to do that today. I've reviewed some of his videos, and he and I really have to talk about consciousness and spirituality and a whole bunch of other things. I think I'm going to get along well with this man. So, Mr. Damer. Anyway, so it's just the opposite. This pristine conditions actually make it more difficult for some of these things to happen. But yet they still do that in the laboratory because they're only interested in one part of the puzzle, right?
Yeah, they're trying to isolate variables, or eliminate variables. Whatever they're trying to do, they're just trying to look at one thing in an experiment.
Eliminate variables and find one thing. But when you have a mixture of all of these strange little chemicals, like I pointed out, the xanthines and all of the complex aromatic compounds that you get in the tar, and in these biopools, it turns out that there are many different ways to stabilize things, and it creates an immense number of pathways which we are trying to unwind. So, Neff is teaching real science right now.
That's good. I'm glad somebody is, because gosh.
Yeah, I wonder if he's half in the bag like he was the other night.
He might be.
Either he's half in the bag or he has a problem with slurring for some other reason. Stroke? I don't know. Not sure.
I wonder if it might be due to excessive alcohol abuse.
Yeah, it does. It does eventually fry your temporal lobes. As my older brother could attest to before he died, that.
He has Will and Andrew. Our rational empiricist.
Andrew again. Andrew.
What's he think he's going to accomplish by talking to Neffy?
He went on there the other night, and it was just horrible what they did to him. I mean, they just ran over him. And now he's back. I love that kid. He's got stamina.
I want to make that point. Yeah, that's true.
He got stamina. So anyway, this is also an interesting little article. So glycine, leucine, lysine, serine all stabilize these membranes. which kind of blows back on James Tour's whole shtick, right?
It's just too hard.
Yep. Yes. That's Cornell. Here's a Deemer paper from Bioenergetics and Life's Origins. Have we looked at that one?
No, I don't think we have.
Okay, this one is from 2015.
Because the only paper we've actually reviewed, now of course you might mean like on the weather report, but is the cell really a machine? And then this hot springs hypothesis. So there is just a world of papers we could review just in support of hot springs hypothesis, much less the other hypotheses that are out there.
Yeah. And I want to mention again that this just works. In fact, Demeter and his partner got the idea from Damer and Dahmer. So that's what. So they very
Damer and Dahmer.
Damer and Dahmer. Is that what I said? Okay, I'm sorry.
You said Damer and Dahmer, which are two alternate attempts to pronounce the same guy's name.
I do that occasionally. Just how my brain works. You see, I think outside the box.
That's right.
You guys are in the box. I'm outside the box.
No, I burned the box.
I'm romping around outside the box and no telling what comes out of my mouth. Anyway, Bioenergetics and Life's Origins from 2015. Central to our understanding of living systems, yet attracted relatively little attention in the origins of life. Bioenergetics. In a second, what's he on about here? Let's see what he's on about. Now there's some new terms: photolithotrophy and chemolithotrophy. Orge, is he out there? He's probably getting excited if he's out there.
Yeah, there's never a geologist around when you need one.
How did ATP emerge prebiotically? Anybody want to Google that? Well, ATP is not so difficult, actually.
ATP can be rather difficult to form, actually.
Adenosine?
Adenosine triphosphate.
Well, yeah, ATP, but how about AMP?
Not too hard. ATP was probably a molecule that replaced other molecules that act as an energy carrying molecule. There's just as many other molecules that work just fine.
Yeah, ADP and ATP are kind of up the energy hill, right? A little bit. Yeah.
It's hard to form because it doesn't want to attach more phosphodiesters to it,
which would require some sort of protoenzyme or ribozyme to accomplish that. And
there has to be something that lowers the activation energy, and the only way, well,
I think that's what this paper is about.
Yeah, that'd be interesting, but it could be that it evolved later on. But either way, no ATP, no life, not necessarily. There's other ways to achieve energy processes.
Yeah, but that one is so prolific that it had to be there at the very beginning. There was something that preferred ATP because it is prolific in life, ubiquitous, thermal energy sort of thing. And I'm guessing that there is a pathway, but Otangelo, you gotta go Google that, or we could Google. We could Google how hard it is to form ATP instead of Googling how did it happen, right?
Yeah, for cells, it's not that difficult because they utilize proton gradients to achieve that process. But hard to say if a similar process would have existed near the origin of life. It might have.
Prebiotic synthesis. Yeah, this covers just a whole bunch of different possibilities, different ways that it could have happened. So this is Deemer 2010, and I should toss the whole list here into the description. But You guys, of course, have all these papers downloaded too, right? Because you guys did all the. You backed me up on this shit, right?
Nope.
Oh, I didn't download the papers. I'm just trying to understand the hypothesis paper, and then look at the other papers once I figure out what's going on.
Yeah. Well, they all back it up. Black Parker. Okay, I got Parker. Looked at Parker. They mentioned grown. What was that one? Polycyclic aromatic compounds, aromatic hydrocarbons as plausible prebiotic membrane components. Now, there's another idea for us. Still another idea. Application window. Grown. Grown. I don't know what the fuck. How you want to do it. Okay. So this one would be an interesting. Also, Deemer. Got to get around.
It's only 12 pages.
Yeah, you see all of this stuff that they call a problem because all these different compounds form are all solutions, not problems. So,
yeah,
I don't know. Why did that bounce off on me? What do we got here? Microscopy and DLS of vesicle solutions. So they've actually done some experimental work with some of this stuff and looked at these. I was hoping for some pretty molecule pictures here somewhere. You know how I like pretty molecule pictures. I don't see any. Dang me. Oh, yeah. Here's some pretty molecule names. Chrysene quinone. Ooh. Chrysene quinone? I love these things. This is the tar that they're talking about. That's why I got excited when they mentioned tar. It just forms a tar. It's like, yeah,
tar.
That's exactly what we need. Tar.
We need more tar.
And brains.
Brains.
Okay, guys. Where do you want to go?
Well, I guess maybe we can, do you want to have some people hop in and talk, or do you want to keep this as a discrete video so people don't get confused where the paper coverage is, assuming people are going to watch the paper coverage, the paper trail?
We can haul people in. Well, the problem is that I have to re-upload these things, and then they end up in, well, I guess that's not a problem. I can split it and re-upload it. We can invite people in if we want to discuss this. What do you think?
So you're going to contact Damer today? Send him an email?
I'm going to try. So Jerry, I'll have to reach out to Jerry and find out where that conference is that we got invited to and let you guys know. But I will open this up. But I may not be here that long. But anyway.
I'm probably going to get going, actually. Maybe we can do an after show later tonight.
Yeah, okay. We can do that. Yeah, let's do that. So anybody that's interested, we'll do an after show on this later, and we'll try to keep this one concise.
Because now you put out the stream. SpringerLink.
So we did our job. So next time, what are we on? Part 5, section 5. We're moving
and then after
on.
that's the conclusion. So
oh, cool. So we could go right into the conclusion. So we have one more paper.
Yeah. That sounds great.
Yep. Goodbye, cruel world.
Okay, goodbye, cruel world. Jerry, did I get an email on that Zoom? Oh, I did get an email, so I do know where it is. Goodbye, cruel world. We're in the broadcast.